Okay, today I want to talk to you about differentiation and stem cells. And so we know most life on the planet consists of one-celled or unicellular organisms. If you can look at the graphic at the bottom, you can see the bacteria in purple. Those are all unicellular. You can see the archaebacteria in red, and those are all unicellular. And you can see the eukaryota in brown, and most of those are unicellular, with the exception of the 3 that I've circled in green. And really being able to turn genes on and off allowed the change to multicellular creatures. And that's a pretty special thing. Humans, being multicellular creatures, actually do start as one cell, and that one cell is going to undergo many rounds of mitosis until you have about 100 trillion cells in your body. So that one cell has to really turn on into all of the different parts of you. We call that process differentiation, which is the same as specialization. So you begin as a clump of cells, which is a blastocyst, and those cells are blank canvases, and they can become any kind of cell. And since all of your cells contain the same DNA, specialization is done by turning parts of the DNA on and turning other parts of the DNA off. So before cells are specialized, we call them stem cells. And stem cells really are cells that are able to differentiate. They're really important for embryonic development, but we also use them later in life to heal injured tissues or to replenish cells. So your body needs to make more blood cells, but blood cells can't make more blood cells, so we actually use stem cells to make more blood cells. Pretty interesting, huh? So we got 3 terms to describe stem cells. The first one is called totipotent, and those are cells that can differentiate into any type of cell. So they have the potential really to stay and develop into an entire organism. We can take those 4 cells in the lower right-hand corner, we can take those and we can do what's called embryo splitting, and we could split them into 4 individuals. So we would have 4 identical quintuplets out of that one egg. So zygotes, which is just the word after the sperm and the egg meet, are totipotent, and the cells really remain totipotent for about 4 days after fertilization in humans. And after about 4 days, the cells start to specialize. We also have stem cells that are called pluripotent, and pluripotent stem cells can differentiate into most cell types. They couldn't give you a whole new organism, but they usually can fall into one of the pathways. They can turn into stomach, intestines, and lungs, or they can turn into muscles, bone, or reproductive tissue, or they can turn into skin and nerves. So it's kind of interesting that those particular tissues are related to one another, right? The last term that we have is called a progenitor cell, and progenitor cells are committed to becoming a certain type of cell, but they really need to mature or specialize a little bit more. So the progenitor cells in your bone marrow only give rise to red blood cells, right? And the red blood cells have a very specific function. How do stem cells know what to become? Well, during development, we're producing different hormones, right? And the hormones switch on and off the genes, and the different signals can cause different chain reactions to be activated or deactivated. And those chain reactions activate and deactivate certain parts of the DNA. Those genes control what that cell is going to become. So we have 2 different kinds of stem cells, really. We have the embryonics. Well, we really have 3, but we'll get to that in a second. We have embryonic stem cells, which are obtained from a developing embryo, and those can be totipotent or pluripotent. They tend to be rather erratic in the lab. They aren't very predictable, and so they're not actually the preferred way of doing any kind of research on the human body, because the cells are so able to become anything, and we don't know the switches well enough to know what to switch on and what to switch off to get them to really behave the way we would want to. So we can't program them well. In the adult or the multipotent stem cells, those are obtained from adults. Those are mostly progenitor cells, but we do have a method that was discovered about 1994 that tells us that we can, I'm sorry, not 1994, 2004, where we can actually induce the pluripotent state in the stem cells, and so we have induced pluripotent cells, and those are reversed from being specialized cells to being pluripotent cells. So we can actually take your skin cells and reverse them back into stem cells, and that's a pretty cool thing. The controversy around stem cells rises from the fact that the original stem cells had to be harvested from an embryo, which meant that we were killing embryos. And really, we weren't killing them. We were using ones that were left over. But people had some really big ethical concerns about, should we be abusing life to harvest stem cells to do research. Some countries still use embryonic stem cells. When we got the pluripotent cells and we could induce the pluripotent state in any cell, that was really a huge breakthrough, and that is what most people use to get stem cells, is that they, right? But the stem cell research still carries